Everybody ready? Yeah, lecture 33. So, overview. These different cell types. Lymphocyte versus neuron. Same DNA there. Then the Gurdon experiment with the frog just won the Nobel Prize. These cells are pluripotent, right? So you can take the epidermal cell and you can generate a whole new organism from it. So it's not different cell types lose DNA, but it's all there, pluripotent in some contexts. Done in mammals, right? Next slide. Transcriptional control, the key regulatory point. See, isn't this fun? Okay, so we just talked about prokaryotic gene transcription, eukaryotic, all kinds of bells and whistles, a huge complex, more than just sigma factor. So this all assembles in a fashion at a Tata box, bent because of the AT minor groove. You've got your complex. That complex then, there's more to it than that. Once elongation proceeds, you need to recruit these other factors because you're going to modify the eukaryotic RNA transcript, right? Cap it, splice it, polyadenylate it. Okay, now, genes are activated at a distance in eukaryotes by enhancer elements. So I didn't know prokaryotes had enhancers. I think that's for experts only, I just think generally they don't, okay? So just wipe that from your mind. They don't have enhancers the way eukaryotes do. Eukaryotes have enhancers. You see the dash, dash, dash. They're not right there. They're hard to find. And then we're going to see later how you find them. And so you got the little green pawn. In this case, we just have one. Later we're going to see there's going to be multiple ones. Okay.
Do we need to understand all those general factors that help initiate transcription? Like
Sarah. Did we? Don't make me. I don't want to understand them. I don't need to understand them. We just know there's a bunch of them, right? Later we're going to see that the helicase or something. But okay. Okay, now, so what else is different about eukaryotes? The chromosomes, the DNA is packed into chromatin, right? And so the promoters aren't accessible. So thus far we see we need all this machinery to assemble at a Tata box. That's necessary but not sufficient. We need the enhancer element. That's still necessary but still not sufficient. So we've got packed chromatin that we've got to open up, right? We've got to recruit. We've got to allow access. And there's two mechanisms, right? Covalent modification of the histones there through an enzyme or remodeling of the complex. Both of those are reversible. Fun. Okay, so nucleosomes. The histones serve as a core to wrap the DNA around twice. Those things can all get packed together. You end up getting a how many fold compaction? 10,000 fold. Enormous, okay? That doesn't happen with the big circular E. coli chromosome or prokaryotic chromosome. All right, and then these are just some pretty pictures and hypothetical mechanisms, how this does it. Heterochromatin. So that stuff is chromatin. It's highly condensed. It stays that way, right? It's not subject to these activation mechanisms, these two activation mechanisms. So that stuff is just inert. One of the X's is completely heterochromatin, and then you can see the effects on activity through these in these two contexts, yeast and flies, showing the effects of heterochromatin on gene expression. Silences it. Okay, so
How does the switch of the gene inactivate it? Like on the white eye?
Yep.
How does it switch?
Oh, it's just because it's say what if these regulatory elements now are in heterochromatin and they're permanently silenced, right? So dead.
How do they get there?
Huh?
How do they get into the heterochromatin? If heterochromatin isn't, if you can't access it, how does stuff get into it?
I don't know what dictates what becomes heterochromatin, but I think when that whole gene moves with its regulatory element, and you have replication.
So you can
Right.
have heterochromatin, you just can't like reopen it up.
It just gets covered, right? So when those yeast cells are dividing, or in the fly eye, yeah. So those nascent cells, then it gets covered and it's silent.
And it can never be opened again.
Yeah. Okay. Okay. So we've got chromatin. It's DNA and protein, and then we learned like the long-standing view was protein had to be the hereditary material, and so then Griffith, the medical officer, showed then by tinkering around with these pathogenic strains that a heat-killed form could transform the non-pathogenic strain. Okay. So he provided this experimental avenue then for much later for Avery McLeod McCarthy to show the DNA was a transforming substance by fractionating it. Okay, so Griffith showed this transformation event, and then the others later showed what it was. Okay, so what are some of the mechanisms then? So eukaryotes, then, we have all this combination of elements. So before we just saw the one green pawn, now we see there's a lot of them. You see the dashes, they can be distributed at great distance, hard to find. But the point is you have multiple inputs through these elements and regulatory proteins. And then the fly embryo in the next slide is just to show how this might work. You know, you can have overlap. So all the cells in this syncytium will have that DNA, but only some of them will have, you'll have these different combinations of, these are all transcription factors, binding to it to give different outputs. Okay, so how do we find where these elements are? Using these reporter constructs, right? So this is where you do this promoter bashing, and you just chop up pieces around the transcription initiation site. These can even be in introns sometimes. So let's just keep it simple. We're just talking about upstream here, okay? And then you put that in front of something that was going to report. Later we're going to, so this is beta-galactosidase, we're going to see GFP, right? So in this case then, it'll produce beta-galactosidase. You use a chromogenic substrate when hydrolyzed makes blue, and that tells you then that that element that you put upstream there from that promoter is responsible for that stripe 2. Okay? So this is done again and again. This is a very powerful tool. All right, so that, hello? Is that you, Carl?
Yeah, I was just asking her, but I'll ask you. So you're saying that you test something out from far away and you bring it closer and that activates it?
No, you don't have to bring it closer. You're just dissecting them. It's not even closer, is it, in this little cartoon? It's even
So you have all
further.
the others.
Yeah, so it doesn't really matter, right? Especially the way, see how these enhancers thing loops back around?
Mm-hmm.
So all you're asking, you're just dissecting it.
So you're just doing individual ones?
Yeah, you just systematically go through and chop it, put it there, and then test it. That's what grad students are for. It's just this mind-numbing, yeah.
Dr. Groppe, question.
Joe?
This is just a curiosity question. It says 3, 2, and 7. Is there 7 stripes? Would there be 7 promoters for this, or did the combination of those two?
I don't remember. I remember when this work was done. A lot of this was done in Switzerland, the lab next door. So this is even skip, the transcription factor. So these